So it will be a task for you and me to keep you awake after lunch. Okay. So, our कौन सा mic है यार इसका? Hello, are you? Yeah, it's fine now. So, I hope you had a good session on microRNAs. So, microRNA is one thing that we really work on in our lab also, and along with other non-coding RNAs. So you have studied bioinformatics in your syllabus also, I guess so, and you know basics of bioinformatics. Okay, so what is bioinformatics? Yeah, I am teaching them bioinformatics because that's why they are able to answer very good definition of bioinformatics. Okay, and that is acceptable if you write that. Okay. So my students, they have already seen some of these slides. Okay. So everything started with this thing. So what is this? This is the discovery of DNA structure, not the DNA as a genetic material. Okay. So we know that the pioneers, Watson and Crick and Rosalind Franklin, they discovered DNA that has the task, but it serve as a store of information that passed from one generation to another, and another is it directs the synthesis of proteins. Okay, we have genes and non-coding DNA, and that carry information passed on to next generation, and then they also perform the functions in the cell, and it is a storage of information, and information is stored in 4 basics: adenine, cytosine, thymine, and guanine. Okay. Now, like we have only 4 bases, but the arrangement of these 4 bases is very particular, that each part of DNA is different from each other, and every organism, every single genus, they are different from other genuses. Even the same species, they have variations. Okay, they are not similar because of the patterns of arrangement of ACGT. So you can imagine how many permutation combination might be there, that they are storing huge amount of information, and still it is not similar in all the organisms. Okay, so that kind of information you get, okay, and that is translated into proteins. Like for here you have examples of 3 genes. So one portion is making skin cells, another is RBCs, and third one is controlling the color of the eyes. So everything is dependent upon the pattern of these 4 nucleotides. Okay. Now, why to sequence the DNA? Because it is the information that is stored in this, and the pattern is important, that how the ACGTs they are arranged. That's why we want to sequence the DNA. That is one thing. Okay, you have studied many sequencing technologies nowadays. Like I told in the morning that we started doing Illumina from almost 17, 18 years back, and then Sanger is also there from 30, 40 years or more than that. So if you want to really come to a conclusion that what DNA is doing, you have to have a sequence of DNA with you, and that sequence you can obtain by using many sequencing technologies. And we have these pioneers, Gilbert and Sanger, who independently developed 2 different methods of sequencing. Okay, in the same year 1977. So Gilbert was suggesting enzymatic degradation of the DNA, and Sanger was suggesting dideoxy chain termination based method of the DNA sequencing. And Sanger is still very common because of its easiness to do, availability of the machines. Degradation method made by Gilbert was slightly complicated, was not much popular, and no one can use it. No one is using that now. But both of them, they got the Nobel Prize in the chemistry in 1980s. Okay, so what happens in this? You have, like in Sanger sequencing, you have a dideoxy that when this dideoxy is synthesized or entered into the chain, it stops the addition of further nucleotides, and you can run on the gel or in the machine and you can see the patterns like this. And we get the chromatograms like this and we can read them what actually is there. Okay. But there was problems in Sanger sequencing because it was low throughput, that you have to take lot of time because it sequence only 500, 600 bases in a stretch. So if you have, like now we have 96 capillary sequencers, 96 samples can be sequenced at a time, but still it takes lot of time. Okay, lot of money is involved in that. And then inconsistent base quality means sometimes the peaks they mingle with each other and don't have quality data. Okay, we have to recheck the chromatogram and correct the basis. Then it's expensive, still it is very expensive, and not quantitative, means you cannot quantify the expression that we have liberty to do in the Illumina or other high throughput sequencing platforms. So these are some of the genome sizes of some organisms compared to human beings. Okay, human and mouse, they have a similar genome in size. Okay, compared to E. coli, they are very smaller. Okay, so why is the genome sequencing important? Like people, earlier they used to prefer work on small number of genes. Okay, from one to few genes of the pathway. Okay, now we have technologies like we want to do high throughput sequencing. Okay, at large scale. Now we have high end computers with that where you can run and analyze the data in very quick time. Okay, so everything is high throughput nowadays. Okay, so you need to understand what's going on. Now we know that the genes are the same. Okay, the genes are same in the mice and in the humans. So we want to understand that why mice is a mice and why human is a human. So for this you have to have the genome sequence. Okay. Then you can understand by genome sequencing that how the gene expression is regulated in particular environment. Okay. For example, in summers, winters, in stress, normal condition, in diseased and normal people, cancer, non-cancer. To study human variations, how the humans, they differ from each other. Okay, we have populations like North Indian, South Indian, East Indian. Okay, immediately people recognize us that this man is from North India, this is from South India, these are from East India. Okay, so these variations we have to find out, and how we are related to other organisms. Then how this genome information relates to the development of cancer or susceptibility of the disease. Now we have like medigenomics, where you are sequencing the genomes to study the medical complications in human beings. Okay, you can predict accurately that person will have a disease or not. Okay, quite early before the onset of the diseases. Then you can study the metabolism or the drug response in particular organism, how they will respond to particular kind of drugs. Now then comes this Myers and Phil Green, that they want to make use of two different technologies. One was classical sequencing, and then we have shotgun sequencing methods. So they decided to sequence the human genome on the basis of these two technologies. Okay, So in normal hierarchical sequencing technology, we have the chromosomes, and we make back libraries of each chromosomes, okay, because you know back what are backs and yaks, okay. So because they can take large amount of DNA fragment inside, 50 to 100 kb very easily. So then you sequence these fragments, and then you can relate these fragments from which back they are coming up, and then you can locate the chromosomes, from which chromosomes they are coming up, okay. But in short-term sequencing, you simply take the DNA, fragment it and sequence it, okay, and then you find out that on which portion these small reads they are matching, okay. So this is time consuming method, okay, because you have to make clones and then sequences, then align, and okay, but you get very clear picture that from where your library is originated and what is on which chromosome, that which gene is on which chromosome. So here you can sequence it very easily, but afterwards you have to do that which fragment is which from which part of the DNA, okay. And then comes the real bioinformatics, when you combine these small fragments and make a larger genome assembly, okay. Now we have many genome sequencing projects that has been initiated and completed. Okay, India has started one genome per day project. DBT has launched that. So we are sequencing small organisms and sequencing every day bacteria, okay, viruses. They will shift to higher organisms also, fungi or other organisms. Okay. And then now people, they are more interested in getting their genomes sequenced. So many people are there who have their genomes sequenced, okay, like this one, okay, and this is Craig's, okay. So they sequence their genomes. Now we are making the gene chips for the genome chips for the individuals. Okay. now we have next generation sequencing technologies, like we have 454, Illumina, Solexa. Okay, then we have what do you call PacBio, okay, nanopore sequencing. So these are second and third generation sequencing methods, or next generation sequencing methods, and they can sequence the human genome in really quick time. If you have everything in your lab, you can sequence a human genome in 2 days compared to 10 years that took a normal sequencing method. So these next generation sequencing methods, they are modern, high throughput, massive, parallel, okay, rapid, price is reduced, okay, time is saved, then you have low complexity, but you have still high error rate that you have to clear out, okay. But it increases the data quantity and quality. So you have millions and millions of read in your data, so you can align them and reduce the error rate. Read length is small compared to the Sanger, because Sanger can sequence 600, 700, 800 bases in a stretch. But these sequencing technologies like 454 used to sequence 500 bases. Illumina nowadays can sequence up to 300 bases. PacBio can sequence 5,000 bases in a stretch, that is very huge. Okay, and the small bacteria or viruses, they can be sequenced in a single shot in the Pacific Biosciences. Okay, so we have like now tools where you call it third generation sequencing, where they don't need any library. You simply use the DNA as a template, and as the DNA passes through the detector, it keeps on sequencing without the need of making library or amplification of the DNA. So NGS is changing biological research and numerous applications. We have seen during the COVID time that we have lots and lots of sequencing of COVID virus, okay, so that we can identify the mutant strains very easily. Okay. So COVID sequencing projects were going on at many places. Okay. So we are planning to sequence anything that is living or dead, that can be done. People, they are doing like conservation cloning, they are trying to revive the mammoth, okay, woolly mammoth and other things. It's like PCR NGS is becoming, and I asked my student to learn this technology because most of the people, they are doing this nowadays, and it is not a wholly technology. You have to be used as a part of this. Okay, you have genomics, proteomics, metabolomics, everything is interconnected nowadays. So you cannot rely just on transcriptome data or genome data. You have to have metabolome data and also the proteome data. Okay, so it is enabling technology, like we have second generation, third generation, and people, they are working on the next generations, that fourth generation may be coming up. So like computers, they are coming up, and we can sequence like 100 bacteria in 4 days or less than a time if you have libraries here available, and starting from DNA isolation to sequencing, we can sequence human genome within 1 week of time. Okay, then we have, like I told you, that Government of India, they have started that bacterial and microbial sequencing projects, either de novo or resequencing. Okay, de novo is when you don't have a reference sequence, you want to sequence it. Okay, then in resequencing, you have a template sequence available, and you just sequence the strains and align with them. For example, we have human genome that will be used as a template. If I want to sequence my genome, I will sequence it and then aligned with the already available genome that is available in the databases, and then how I will figure out how much I am different from the genome that is available in the database. Then I can detect the mutations, deletions, insertions, SNPs compared to the reference genome. Then this will help in like personalized medicine is upcoming field where you can design your drugs. Okay, for individuals, nobody will be taking the same medicine again and again that is given to hundreds of people, because the response is totally different in different organism basis on their genetic architecture. So you will be given the dose that is essential for your health. Okay, not overdose or lower dose. Then disease inheritance studies, that how the diseases they are passes on, and then we have transcriptomics, RNAs, uh, microarray sequencing, and nowadays we have many, and you have a lecture on small RNAs in the morning, so they do deep sequencing to isolate microRNAs or other small nuclear RNAs. So for genomics, you start from the DNA. To take the DNA, then make the small fragments of the DNA, then make the libraries and then sequence it and then realign them with the chromosomes. Okay. So you have a DNA, you can do like de novo genome sequencing, then resequencing, for example, chromatin immunoprecipitation is there. So to study DNA, RNA, DNA protein interactions, methylome studies are there to study methylation patterns, then amplicon sequencing to study the microorganisms present like we do in the soil in our case, and you can do it everywhere. For metagenomics, then for variation dissections by SNPs or microarrays or through microsatellites, you can do that. Then in the transcriptomics, you have RNA as the starting material where you can isolate the RNA, then mRNA if you are typically targeting the genes, and then you can make library of RNAs, and then you can make sequence out of that, analyze the reads. But you can now, we do whole RNA sequencing so that we are interested in other aspects also, like we have microRNAs, non-coding RNAs, 15 different kinds of RNAs are there, so that are located on different genomic portions. So instead of nowadays sequencing mRNA, people prefer to sequence all the RNA that you got. Of course, there will be high population of 28S or 16S or other ribosomal RNAs, but small portion of others will be there that you have to identify. So for RNA or transcriptomics, then you can do RNA sequencing, whole transcriptome, shotgun sequencing, then you can detect SNPs within the genes, then RNA species other than mRNA, and quantitative sequencing without normalization. So you can compare the expression. Earlier we used to have like Nathan blotting to compare the expression. Then we have microarrays. Okay, microarrays were there. Then we have real-time PCR, and now we are combining this technology along with the real-time PCR. Okay, microarrays like absolute technology. No one is using that nowadays. Then discovering non-coding RNAs that you learned in the morning. These are very important. They are controlling many different things, because I said that all the genes in all organisms, they are similar. Okay, some of our genes, they are highly similar to even very lower or primitive organisms. So if the genes are similar, their working is similar, we have the same pathways starting from glycolysis to amino acid synthesis. So the things that are making different us from others might be located in the genomic regions, and these things, they are controlled by non-coding RNAs. So you have miRNAs, siRNAs, pvRNAs, racRNAs, tacRNAs, long non-coding RNAs, and any other kind of RNAs that are there. So they are encoded in the genome, so they have their own genes, they are transcribed. Okay, so more than 90% of our genome is transcribed, only 2 to 3% is translated. Okay, so we are only, earlier we are only focused on micro mRNAs that are only the 5% of the, less than 5% of the genome, 5% of the RNA. Okay, but now we are looking at the larger picture, that why entire genome is transcribed. Okay, so what the transcription of entire genome is doing. So now we know that they are making many other things that are not making proteins, but they are regulating the expression of other genes. Okay, then we have metagenomics. Like we don't know much about microbes yet. Only we say that less than 5% is cultured in the labs, more than 95% they are still non-culturable. Okay, so if you encounter this, you have metagenomics, where you can directly isolate RNA from any particular niche area and then sequence it, and you may get some information about the species present there. Okay, so for metagenomics, you can choose any ecosystem starting from like oil spills, sea, take workshops, air, water. Okay, so that can be used to do metagenomics and compare with the known. Okay, so we have different sequencing methods, like sequencing by synthesis, like Sanger's giant deoxy chain termination method was there. So they were synthesizing the DNA and then sequencing the DNA. So same kind of technology, where pyro sequencing was there, then Illumina used reversible terminators, then ion proton semiconductor in the solid platform launched by Life Technologies, but it's not very common nowadays. Then PacBio use zero mode waveguide. Okay, then sequencing by oligoligation, again solid is there. Then we have Helios by Heliscope. So these are there, but most common out of these is, earlier we have 454 and Illumina, but nowadays we have Illumina and Pacific Biosciences or PEC Biosciences. Okay, so these are two very common nowadays. The machines are very simple, not very big one. Like you have a gel dock machine, so similar kind of machines are there. Okay, so you can use them. And so we have on the basis of Sanger's, we have sequencers like from ABI or from nowadays another company, Promega has launched their sequencer. So when you put your sequences in that, and they give you chromatogram like this. So other technologies, they also give you chromatograms like this, but they are slightly different. So this is like ligation based method from solid, where you have extension like in PCR. So with every base you have a different dye attached. So when it attaches, it emits light that is detected by the detector, and you have the sequence in your hand. So this is how the Illumina works. So you have uh, adapters that are based on a plate. So you attach your DNA with the adapter, and you have amplification in the form of a bridge PCR. So you have localized clusters of the DNA you want to amplify, and each cluster is amplified by the amplified and sequenced by the technology. Okay. So this is how it is working, sequencing by synthesis. Okay. Again, you just extend the DNA, and this is pyrosequencing. It's called a bead PCR. So instead of plate, you have a bead on which the adapter is ligated. So your DNA attached to this adapter, and then extension happens, and then you have sequencing of the DNA in the 454 machinery. So all of them, they are very small in size, not very big one. Okay, you can see that difference in their size. So even the 96 well plate is larger than the sequencing platforms. Okay. So whatever you do, you have to start from nucleic acid isolation. Then if you want to work on RNA, you isolate RNA. If you want to work on DNA, you isolate DNA, and then make libraries, and then you have the same platform, okay, same technologies to sequence these. Okay. The main thing in all these platforms is they have different kind of chemistry for sequencing. Okay, the main thing that we want is the read length. Okay, like mentioned here, like PacBio give more than 5,000 bases, Roche gives us more than 500, Illumina gives us more than 300. So with very low read count, you have to spend more and more time on computer and check for errors, reduce errors. So you will have large amount of data, but most of them will not be of good quality. So you have to eliminate that data. So we want long reads with low error rate and low price. So price is almost similar compared to the Sanger. It's very, very high. Okay. So this is the price rate of different technologies, where you have less than $1, you have complete genome, and in Sanger's method, it's very, very costly compared to this one. Okay. So if you want to compare the prices, then if you see, when we sequence the human genome, it cost us $2.7 trillion, and it took 11 years to complete that project. The project was launched in 1990, sorry, 1990. It was about to complete in 2003, but because of that sequencing race, it was announced in 2001 only. Okay, then in the same year, another company by Craig Venter, Celera, they sequenced the human genome in $100 million in 3 years, because they were using the shotgun sequencing method and they were using the normal hierarchical sequencing methods. So the time taken by them and the price, there is huge difference in that. Then this technology became commercial in somewhere in 2005 and 6, and at that time 454 was able to sequence the human genome in $1 million in 3 months. And then appeared Solid, they take $60,000 and they claim that they can sequence human genome in 2 weeks. And then comes Illumina, that was a game changer, and they further reduced the price, and now at the current time you can sequence entire human genome in less than $500. Okay, even in India we can do that. Okay, so the race is still going on. We want still better, cheaper technologies, faster technologies. Okay, but for that you have to pay some price. Okay, error rates are more, but this SMRT PacBio, this is a very useful technology when you are working with microbes, small genomes, because it gives you results in very quick time and long stretch of DNA. You hardly need any assembly or other kind of analysis that are required for Illumina or others. Then we have nanopores, sequencing and other sequencing methods that don't need any kind of library preparation. The DNA passes through the detector and with each base they detected. So they also save a lot of time and money. Now the things comes, we call the bioinformatics, so we know the ACGT pattern, but how we can analyze that? That's the biggest question. So in more than 22 years, we are not able to analyze the human genome in quite a detail. So we are still lacking in complete analysis of the human genome, and we need more and more good bioinformaticians. Okay. So bioinformaticians are there in India, but they work for large companies. So in the universities, we really need those people who are very competent to this kind of analysis. Okay. So the things that we require after this sequencing is data analysis, data assembly, and data annotation. Okay. So you have generated the data, you have to assemble it, assign them the location on the DNA or on the chromosomes. Okay. Because you have gathered a small fragments. Okay. Then you have to assemble them to make the large fragments out of these, and then you have to do annotation. Annotation is assigning the functions to the genes that or to the non-coding RNA, DNA, that what is it doing in the DNA. So you annotate that this is a coding region of the DNA, this is a non-coding region. If it is a coding region, then what is the composition of the gene, how many introns it has, how many exons it has, where is the poly A signal, what kind of promoter it has. So everything you have to annotate. Okay. So this is the guide map, how you filter that. So we will discuss that guide map in the coming days. Now we have some reference sequences. Okay, like we have NCBI database. So this was the first database created by the Margaret Dayoff, called Atlas of Protein Sequences and Structures in 1965. The protein sequences started very early compared to the DNA, but the technology was not so advanced. So still we are lagging very much behind in sequencing of the proteins. Still we are able to sequence only 10 to 15 amino acids. Along with that, we have many other databases that are used for different purposes. Now, how can you annotate the genomic data? That will be the things we want to understand. Okay. Any question till there? All are awake or? Okay. So we have different tools for gene annotation. Okay. You are all familiar with NCBI, BLAST? Yes or no? Okay. So who are in graduation, they might not be familiar with that. Anyone from graduation? BSc? From which department? Biotechnology. So don't worry, I will be here for you. I will teach you what is BLAST. Okay. So when you want to annotate the genes, you can use BLAST as a tool, but we have specialized programs available like GRAIL, Expression, GenScan, FGene, FGenes, GenMark. They are there that can find out the genes from the genomic DNA. Okay. And it's not very easy to find out genes. In BLAST it's very simple. You simply take the fragment of DNA, put it and run the BLAST, and you will get the results that this sequence is matching with these sequence in a database. But when you run FGenes or FGene, you're given like 100 KB stretch, and if you have only one or two genes in that 100 KB stretch, it will be very tough to find, and for them also. So these programs, they're highly specialized and trained to detect the genes in the DNA, starting from the exons to the introns and other characteristics of the genes. And we have many expression databases like ESTs are there from humans and many other organisms. And Unigene database is discontinued now, but we have other reference database that are available. Okay. Then Ensemble is there. Ensemble is the largest repository of genome from plants and animals. So we have Ensemble plant and Ensemble animal. Along with the genome data, you will also have the transcriptome data, and annotation of the data is also available there. Okay. Then for protein data, you have different protein databases. Okay, non-redundant protein database are there, like SwissProt is there, PDB is there. Okay, then we have UniProt. Okay, from Unigene it was translated. So now they have changed it to some other thing. Then we have the database of protein families and protein motifs. So we have many databases for proteins for different functions. Then we have additional databases for promoters. Okay, like for microRNAs you have database, for long non-coding RNAs you have database. Okay, so we have database for every specific feature of animals. You have like database of Mendelian inheritance in man for some specific sequences, like telomeres database is there. Okay, so you can search all these database depending upon your requirement. Okay. So we have NCBI genome database also. So who are more familiar with NCBI, they just click the genomes and they will give you how many genomes are there, and you can find out the genome. Similarly, you can go to Ensemble, you can go to the human genome database or a Tiger, or we have hundreds of databases available. Okay, so you can search chromosome-wise gene sequences of humans, their SNPs, their other DNA markers. Then we have different databases located at different places, like this is at University of California, Santa Cruz. They have their own genome browsing database. Okay, everyone who works in like bioinformatics, they always want to create their own database. Then we have proteome database. So if you have a genome and proteome and transcriptome, it's very easy to annotate everything. Okay, so you can directly tell that these are the protein coding regions in the DNA, and this is the gene. Gene will be intronless. When you will map to the DNA, you can find out the intron and exon positions very easily over there. Then you have, like at NCBI also, I told my student that at NCBI also you have many things to read. Okay, you have lot of literature available at NCBI. For every program they have specific tutorials. You can read that and you can be a bioinformatician. But you have to have the basic computer knowledge, how to run the programs. Then Ensemble is the largest genome database. So most of us visited this Ensemble. So we have Ensemble plant and Ensemble animal. So wherever you want to go, you can go and download your sequences. Okay. Then you have a genome alignments. Like if you want to compare like human genome with any other genome, so you can do mapping of cross species comparison, like synteny mapping we do in plants, that how the regions they are located in different organisms. So you can do at Ensemble. Okay. So that's all for now. So if you have any question, please ask. Otherwise, we can move to next lecture. I will take another lecture. So that was basic of sequencing, and then we have how to analyze the data. अभी हमने बायोइन्फॉर्मेटिक्स तो पढ़ी नहीं है. Now you have seen this picture in the previous slide. Okay. So when you have lots and lot of data, what to do with data? Store it for what? For next person to come and analyze? Yeah. Because we know that the information generation is one thing, converting the information into knowledge is a totally different thing. Okay. So in the first lecture, we have created the information. Okay. But we have not created any biological sense of that information. Okay. So now we will discuss how you will create the biological sense out of this database. So this is how you typically do annotation when you are sequencing a genome project. Suppose you have sequenced a genome maybe from any source, fungi, animal, plant or anything. Okay. So you have used a technology like Illumina or Roche where you have ligated the adapters, sequenced it, and after that you have to assemble the data. Like I said, you got small reads in your data. Okay, for Illumina you have 200, after filtering 250 maximum. Okay, so if you put that 250 into BLAST, that will not give any significant result. Okay, unless and until it matches the criteria set on the BLAST. So you have to generate larger contigs out of that, and you have to assemble that short reads into larger fragments. That's how we do this. That we have, suppose these are two reads, and you overlap this, then you overlap this, to overlap this, and you overlap this, and that's how by matching these short reads and overlapping with each other, we generate the larger contigs and assemble the genome data. Okay, and then suppose in a stretch you sequence 100 million reads. Okay, that's the average number of reads what we get after Illumina genome sequencing, 100 million reads. So 100 million reads into 23 chromosomes or 22 chromosomes that we want to check it. फोन सुनना अलाउड है यार इसमें, वीसी ऑफिस से आ रहा है, फोन सुनना अलाउड है कि नहीं? ये बैक कॉल करना, वीसी ऑफिस से है, पूछ लेना. So after you assemble, you finish it, and then you annotate the data and you release the data, public release the data. Okay. So these are the steps of genome annotation project. So our goal is to completely restore the genome. Okay, so that no information is lost during these steps. Okay, and produced high quality consensus sequence. Now, no data is lost, what it means? Okay, so they say that if you have sequenced, like we have a 3 trillion okay, कहाँ पर? So suppose you have, like we know that we have 3 trillion sequences in the human genome. So what it means that we don't want to lose any information. So suppose you have generated a data that is very high quality data, then you are able to retrieve 3 trillion out of 3 trillion sequences. Okay. So when you have a low quality data, then might be you will not be able to retrieve all 3 trillion sequences, reads. Okay, you may get 2 trillion, 2.5 trillion, or you want to save some money, कि आधा अधूरा data मुझे दे दो, okay, 1.5 trillion. That means you don't have a quality genome data. You don't want to lose anything. Okay, so you want the complete data set out of that. Okay, Suppose in normal cases we would do PCR. So in PCR, if we lose one or two bases, okay, by either by sequencing or by error in the DNA polymerase or by some other chance, that 2-3 bases will make a huge difference, because they will totally change the protein, the structure, the function, and maybe they will not make the protein because you have accidentally got an stop codon in between that because of that error rate. So you don't want any error rate in that. So we want everything very high quality. Okay. So whenever you do sequencing or transcriptomic data analysis, don't compromise on the quality. Don't try to save some amount of money. Okay. That because that some amount will cost you a lot. So we have already discussed that you have to choose a genome workflow, extract DNA or RNA, depending on what you want to do. You want to do genome sequencing or transcriptome sequencing. Choose best sequence technology for that project that is available in the market. Like we use Illumina. I don't know why, but we trust Illumina a lot. Okay, that's not the vendor, but the Illumina technology. Okay, then do sequencing, and then do quality assessment and other pre-assembly investigations. Okay, so we have a girl in my lab, Rivi. Okay, and many of our students, they know how to do this quality assessment of the data. Okay, so in the beginning you come to know that your data is of good quality or not after looking at the sequence. Okay, because sequencing companies sometimes they simply make a DNA sequence for you. That's what we recently got. They just manufactured a sequence for us. That was not a real sequence. Then we said no, we sent them back that this is not a sequence. Okay. Then other pre-assembly investigations. What are the other these pre-assembly investigations? That you want to know that your data is ideal one, and instead of analyzing it further, you want to check whether we should proceed or not, because there are many steps. Like for example, if you want to do transcriptome blast assembly, then it will take 15-20 days to complete that. Okay, so after the blast run, you come to know that your data is not good. So instead of waiting for that 20 days, in the very beginning you check the quality of your data so that you decide whether you want to proceed or not. Okay, then after quality assessment, you do assembly, and we have many assemblers nowadays. Most commonly, because who do Illumina sequencing, they use the Trinity assembler that is given by the Illumina. Okay, then you do assembly validation. Like we are only using the computers to validate the assembly. Then how you come to know that the assembly you have made is a good quality assembly, or you simply, okay, good quality assembly means you are combining step by step. Okay, so you are sure that this fragment will come after this fragment will come after this, and in this such you will get this fragment. So what happens if one is matching like this, another is there, so there will be a lot of hodgepodge in the data. So that means the assembly is not a good quality. So you have to validate the assembly also. So for this you have to generate some of the cDNA library data from your genome or from your transcriptome. That will tell you that your assembly is good. Then for this you do assembly comparison. We have many assembly tools, and you compare them with each other. I always tell students that always use different tools. Never trust on one tool or one program. Okay, because one program may be given some kind of false results, and you want a repeat masking or not. Like we have many repetitive elements in the DNA. If you are doing transcriptomics, then you can easily mask the repeats. Okay, you can eliminate the repeats from your transcriptome data. But if you are doing a genome assembly, then you have to think, because we have very large repetitive elements present in our DNA, how to tackle that repeated elements of the genome. And then after doing all this, you finally go for annotations. We have many programs for annotations also. Now, for doing the assembly, you should understand the assembly process. Then coverage of the data. Coverage of the data is that how much data you have covered and how many number of reads per fragment will be present so that you can get a good coverage of the data. Okay. Suppose I have a DNA sequence, I said I want 100x coverage, means I will have 100 reads from the same template. Okay, so I have 100 from here, 100 from here, 100 from here. So if I said only one read or two read or five read or 10 read, then we may have some problem because these sequencing technologies, they have very high error rate. So when you will increase the coverage, that means then you will be able to make a good assembly and reduce the error rate. Then you want to do paired end sequencing and mate pair sequencing. Okay, like paired end when you sequence from both sides. So you have a DNA fragment, so you sequence from this side and you sequence from this side. Okay, suppose you have a clone where the fragment size is 1 kb and you are using Illumina sequencing. So you have sequenced 300 from this side and 300 from this side. So total is 600 and you have 400 bases that are left. So how you will sequence that? You will make a mate pair library. So you will invert the sequence. Okay. So the inner one will go outside and the outside will come inside. So that is our mate pair libraries. And then you have to check the insert size. So depending on the technology you are working, you have to define the insert size. For Illumina, we keep it 300 to 500 bases. For 454, it can be 500 to 1 kb because these different technologies, they can sequence different lengths. Okay. Then file formats, they are different. We know only FASTA format. The FASTA format is there, but they generate FASTQ format. So you have to know how to interplay with the formats. Then contigs and scaffolds are there. Then N50 values, they are there, and that you have idea for that. So in the next generation sequencing, we first fragment the genomic DNA and sequence it, and millions of small reads, they are generated. Like I said, 100 million on an average we produce for an average genome. Okay. So depending upon the sequence technology, it may be up to 100 base pair or 100 kb in length. Okay. So these are different technologies where in Illumina we make these clusters and sequence it. Okay, and then in the mate pair libraries you break from the fragment and invert that. Okay, but aim is to get complete information out of these fragments. Okay. So in the paired end reads, so the fragments, they are sequenced from the ends, in the mate pairs from the center to the opposite ends, and this tells you the number of reads, the coverage. So this is 1x, 2x, okay, यहाँ पर 3x, 4x, so if you 5x, 6x, 7x, 8x, so depending on the number of reads you have for fragment, it will be multiplied accordingly, 1x, 2x, 3x, okay. Then you get the output in this format. This is called FASTQ format. So you have numbers written here, okay, not starting with greater than sign, it's starting with @ sign. So @ sign, it's a FASTQ format file, and then you have your sequence written here and then followed by some of these words. Okay. So this tells you which instrument you have used, which run number, which flow cell ID, which lane, the title of sequences, position X position, Y position, read, it's a single end read or paired end read, output आपको कैसी चाहिए, everything is mentioned in this first line, command line. Then you have sequence, and this line tells you about the quality of the data. Okay, that how accurate is the base calling, that how confidently the system is saying that this base call was good. Okay. So for paired end reads, you will get, like here it is written from one side one end and from the other side it's other side. So you will get that. So you can filter out one for one side ends and two for two end ends. So when you are assembling it, so because they are running anti-parallel to each other, towards each other, so one side will be in reverse order. So when you say paired end, so it has to be reverse complemented to making the assembly. Okay, so if you will do like this, then you will never get the assembly. So paired end is written there, and then everyone is familiar with the FASTA format. So we assemble these short reads into larger ones, and when you assemble these into larger ones, you get the sequences in this format. So suppose you have 100 million reads, you can convert into 1 lakh reads. ठीक है. तो assembly के बाद, if you are assembling a transcriptomes, typically we ask for 50 million reads in transcriptomes, and then we assemble them, and we are left with 100,000 reads. So we are, after assembly, we are reducing a huge number of reads into few thousands. Okay. Then how to assess the quality of the data? Okay, it's calculated by Phred score, that how accurate is your sequencing results. Okay, so Phred score is the score which tells you that how efficient is the sequencing data. Okay, so it gives score for each base. Suppose you have sequenced 100 bases, so it will give you Phred score for each base. Then you will come to know that from where you have to cut the fragment for better assembly. Okay. So how it helps? So it helps in assessment of the sequence quality and recognition and removal of low quality sequences by end clipping, because you have adapters at the end. People who have done sequencing, they know that in the beginning and in the end we have low quality data. So we basically trim the front portion of the sequence and the last portion of the sequence and keep only the middle portion of the sequence. Okay, so this Phred score tell you from where you have to trim the data, and later on you can determine the accurate consensus sequence. If you have a good Phred score value, you have a good trimming value and good overlapping value. So if you have a Phred score of 10, that means you have an error rate of 1 in 10 bases. That's very high. So roughly your 10% data will be inaccurate. Okay, so you have to filter that, remove that data. So if you are talking about 3 trillion reads and you are losing 10% of data, you are remaining with only 2.7. That is a huge amount of data that has been lost. So basically, we accept the Phred score that comes higher than 20. For us, I prefer more than 30. Okay. So if you are choosing a Phred score more than 30, that means you have 99.9% accurate data. Okay. You don't expect 100% accuracy in the data. Okay. So how it tells you that for each base, if anyone has seen the Sanger's chromatogram like this, so with the chromatogram they share some other files, okay, and that tells about the Phred score of these. Okay, so here you have a good peak line telling that Phred score is high. So here it comes down because you have some noise in the data here, and again it starts increasing. The wider is the size of this peak, that means you have good quality Phred score. जितनी वो नीचे को आएगी, that means the Phred score is not very accurate. Okay. Then for doing de novo assembly, so you have fragmented your data into small reads, and you want to combine them together to make a consensus sequence out of that. Okay, so you want to make a consensus out of that. So how you will do that? So once you get the data, you get files like this. So you have small reads like this, and you trimmed the adapters because you have adapters on both the sides, whether you are doing 454 Illumina, on the both sides you have adapters, you trim that, and then you start assembly. So you have many reads that are targeting the same region, so they will locate on, make clusters there, and here you can see that something is missing here, it's star or n, then you can replace the n with, because you have many sequencing that shows that here should not be star or n, you should put G here. So that is the advantage of coverage. So if you have more and more sequences per fragment, then the accuracy rate will be increased. Okay. So that's how you generate overlapping reads. So suppose if you have a reference sequence, like if I want to sequence my genome now, so we have already human genome available, I will simply sequence it and align with the already available genome data. So this will be the available genome, this will be my genome, and they will align with that, and where there will be any insertion, deletion or mutation in my genome, that will be highlighted that this particular fragment is not matching. That's why I come to know that I am different from that person in these areas. Okay. So you try to assemble this, or if you are doing de novo without any reference genome, okay, then what you do, you take the reads, assemble them automatically. Okay, they will automatically overlap with each other depending upon the value you have given. Okay, and then they will make a consensus sequence like this. That will be your draft assembly of the genome. Okay. So you will get this. So when you have more coverage in these regions, okay, so this will be more confident data because you have more coverage in these regions, and here you have less coverage. So this will be a low quality data. Okay, this may be true if there is no error rate, but we cannot say with confidence that this is very high quality data. So you need more reads or more base coverage per data fragment. Okay, so you can see it is 5x coverage, 2x coverage, so we want 100x coverage. Okay, so depending upon the genome size, like how you come to know that I should sequence only 100 million reads or 50 million reads or more than that or less than that. Okay, that will be dependent upon the genome size. What is the genome size of an organism? Like for example, we have 100 MB of Arabidopsis, 1.1 GB of wheat, okay, 500 MB of grapes. We are working with these, that's why I remember these. Okay, so you cannot use the same parameters that you are using with the Arabidopsis with the wheat, because wheat genome is 10 times or more than 10 times larger than the Arabidopsis. Similarly is with Drosophila and humans, or C. elegans with humans. So you have to first check the genome size of your organism and then only go for the read counts. Okay, how many reads you required to get the accurate coverage of the genome data. Okay. So that we already explained. So if you know that this much is my size, I want 50x coverage, I am ordering 120 base billion, and then you can easily calculate that how many reads you will require out of that project. Okay, then insert size. So you have adapters and primers. You will remove it because they will also be sequenced. So they take lot of data. Okay, so you should always include adapter primers. So if you are sequencing 3 trillions, so by adding adapters, it will be more than like 3 trillions. Okay, then read 1 and 2. So this will be your insert size. So this is inner may different, that we just explained, that you have 600 bases and in between you have left. Then you break the portion from between, okay, and then invert it and get the dividend. Then what are contigs and scaffolds? So contigs is when when you combine the small reads and make a larger stretch of DNA. So that is called the contigs. So what is scaffold? And several contigs, they stitch together with n between them. So you know that, like, I have two sequences, two contigs that are very large in size. I know that they are part of chromosome 1, one is present here, one is present here. So in between I have a missing data, so I will put there and put n and n's between them. So if you see the chromosome sequence on the NCBI, then you will see many n's written there. Okay, so that n's means the data is missing. So what you can do, if the n number is large, small, you can make primer from one end, and you can try to amplify that. But the PCR has its own limitation, it cannot amplify more than 10 kb fragment. Then you have to cut it, make a library of that, then do sequencing to complete that ants in the scaffold. So that slowly people try to fill those gaps and try to convert these ants into the useful, valuable data. Okay, so if you have contigs, and you can convert it into ants, scaffolds, by putting ants between them. So this will called a scaffold. So in the NCBI, you can see these kind of sequences that are there. Okay. So N50 is the mean value, is the measure of quality of the data again. So N50 means half of the reads they are higher than this, and half of the reads they are lower than this. So if you have a 5,000 sequences, and so say 50,000 sequences, and you have N50 value of 1 kb, that means 25,000 they are larger than 1 kb, and 25,000 they are smaller than 1 kb. Okay. So for genome assemblies, you always want higher N50 value, and that is called NG value. Okay. For transcriptomes, because average size of a gene is 1.2 kb. Okay, so you need N50 value somewhere around 800 to 1 kb or 1.2 kb. It's enough. Okay. So NG is for genomes. We already discussed. Okay. So Illumina, it produce, like, I personally like Illumina very much, so I talk a lot of Illumina. So it produce huge yield, cheap, reliable, read length is long enough. Industry standards, they are very high. They have very high good machines. So they have problem in where you have more GC content, their sequencing has some problem. Then quality dip at the end of the reads, that is with all the technologies. Okay. And then a long running time for high-seq and short insert size. Then we have PacBio that has produced very long reads, single molecules, high error rate are there. So you lose almost 50% of the data because of these error rates. Then we have nanopore sequencing, Minion are there. They are slightly wider than iPhone, length उनकी इनकी होती है nanopore की. Okay, but the error rate is very high. Okay, now the companies, they come to us that please take this nanopore from us. We will give you free of cost, but you can buy chemicals from us. So they will cover the charge of this machine in the chemicals. Okay, but the problem with this is the error rate is very high. Okay. Now that's all for today. I think more than 1 hour is left. You cannot digest more than that, or you are able to digest. No? Okay, so we will continue this in the next class. Any question in this, you can ask. We have not reached till that. Yeah, that was steps. We are just covering some steps. Assembly doing, we have not done any assembly yet. We are just covering the basics of assembly. Okay.
Sir, the thread score we discussed, and you have this quality score given within the FASTQ format. Is this same or different?
Yeah, it's the same. It's calculated on the basis of thread scores only. We have programs to measure the thread scores also. So They also gave us, and we also calculate, because we have QC programs that checks the quality. Okay, because sometimes these companies, they can manipulate with the files.
So
Okay.
there is additional information provided in FASTQ format that we can look for which platform is used, and is there like information provided which kit was used for library preparation?
They have standard kits. But once you send them the sequencing, they tell you everything. That this kit they have made used for sequencing, this was the adapter sequence, how they used it, how they make the library. So they send you everything, that how they have proceeded.
For cross-checking purpose, it is not possible to deduce from the data that we receive.
No, no, no. You just have the adapter sequence. So adapter sequence, they are particular for particular companies. Like Illumina has their own adapters, 454 has their own adapters. So on the basis of that adapters only you can come to know that which company has sequenced that. But that adapters can be used in many kits, okay, within the same company. Okay. Anything else? Nothing? So thank you. So we will meet you tomorrow. Some other fellow will be here. Okay. Thank you very much.